For over a decade, scientists classified Ötzi the Iceman's maternal lineage K1F as extinct. No living match existed in global databases. His paternal haplogroup, GL91, survived in just 19 men across the Tyrolean Alps. Then in 2026, FamilyTreeDNA's MitoTree linked K1F to a living man with maternal roots in the Shawiya Berber community of northeastern Algeria. Ancient DNA from a 5,300-year-old Copper Age mummy now connects to people alive today. But none of this was possible until scientists replaced a contaminated genome that had been distorting Otzi's genetic story for over a decade. On September 19th, 1991, the discovery unfolded when two hikers crossing a high alpine pass near the Similon Peak encountered a human body emerging from melting ice at 3,210 meters. The location, on the border between Austria and Italy, placed the find in the Ötzi Alps. Archaeologists confirmed the remains belonged to a man who died more than 5,000 years ago, during the Copper Age. Radiocarbon analysis of bone and tissue set the date at about 5,300 years before present. The body was naturally preserved by ice, with skin, organs, and even his last meal intact. Alongside the mummy lay a copper axe, a longbow, arrows, stone tools, and leather garments, evidence of advanced metallurgy and textile skills in prehistoric Europe. The South Tyrol Museum of Archaeology in Bolzano was designated as the primary repository for the remains and all associated artifacts. Over the next months, forensic teams cataloged 61 tattoos on his skin, analyzed his clothing and weapons, and collected samples for future scientific study. The find, quickly dubbed Ötzi the Iceman, offered the oldest complete human body ever recovered from Europe. From the outset, researchers recognized the specimen's potential to answer questions about Copper Age diet, technology, health, and migration. The level of preservation made it possible to extract both mitochondrial and nuclear DNA from bone and soft tissue, opening a new chapter in ancient genomics. The scientific community began to treat Otzi as a reference point for prehistoric human biology in the Alpine region. Every detail, from isotopic signatures in his teeth to the pollen grains in his digestive tract, became a data source for reconstructing Copper Age life. The exceptional state of the remains also posed challenges. Preventing modern contamination required strict clean room protocols and careful handling. As the first ancient European mummy with recoverable DNA, Otzi set the standard for future paleogenomic research. The discovery launched a wave of studies seeking to map his ancestry, health, and genetic legacy, establishing a foundation for the genomic investigations that would follow decades later. In 2012, a team led by researchers from Stanford and the European Academy of Bolzano published the first draft genome of Ötzi the Iceman. This analysis was built on DNA extracted from bone powder taken from his left femur, with sequencing performed on an Illumina HiSeq 2000 platform. The resulting genome achieved only 1.3-fold coverage, meaning that, on average, each position in Otzi's genome was read just over once. Such low coverage left many gaps and made it difficult to distinguish genuine ancient DNA from contaminating material. The laboratory conditions at the time did not fully isolate ancient DNA work from modern human DNA, raising the risk of contamination during extraction and library. Preparation. The silica-based protocol lacked partial uracil DNA glycosylase treatment, so post-mortem DNA damage was preserved, but so were undetected modern fragments. Authentic ancient DNA typically appears as short, damaged fragments, but the 2012 dataset included a significant number of longer molecules, over 120 base pairs, characteristic of modern DNA. Bioinformatic checks, such as fragment length analysis and damage profiling, were applied, but the low sequencing depth limited their effectiveness. The contamination could not be reliably measured or filtered out. Instead, the team relied on mitochondrial haplogroup concordance and X-chromosome heterozygosity to estimate purity, but these methods are underpowered at such low coverage, especially for nuclear DNA. No robust nuclear contamination estimator was used at this stage. Population genetic tests, including admixture and F statistics, placed Ötzi on an ancestry axis associated with steppe populations, a result that contradicted archaeological expectations for Copper Age Alpine Europe. Later reanalysis demonstrated that the steppe signal was an artifact. Just a handful of contaminant reads, carrying alleles common in Western Europeans, were enough to bias allele frequency methods when the authentic ancient DNA signal was so sparse. The modern DNA introduced during extraction and library preparation had gone undetected, distorting the ancestry profile. The 2012 genome quickly became a reference point, cited by more than 150 studies. However, its findings rested on data that could not be fully authenticated. The limitations of low coverage and the presence of modern contamination produced a misleading picture of Otzi's ancestry. This left open a fundamental question. What would a high-coverage, contamination-controlled genome reveal about the Iceman's true origins? On August 16, 2023, a new high-coverage genome for Otzi was published in Cell Genomics by K. Wang and colleagues, a DOI 10.1016/J.XGN.2023.100377. The team extracted fresh bone powder from the original femur sample, working inside a dedicated clean room for ancient DNA. The facility used positive pressure air systems, ultraviolet decontamination, and full-body protective clothing to prevent the introduction of modern DNA. All equipment and reagents were sterilized, and extraction blanks were run alongside the sample to monitor for contamination. The DNA extraction method targeted ultrashort, damaged fragments typical of ancient remains. Library preparation included partial uracil DNA glycosylase treatment, called UDG half treatment, which removes most internal deaminations while preserving terminal damage. This damage pattern is a molecular signature of authentic ancient DNA, and it allows reliable authentication of the sequence origin. Sequencing was performed on an Illumina NovaSeq 6000, generating over 140 gigabases of data. The final genome reached an average coverage of 47-fold, meaning each base was read an average of 47 times. This depth enables robust consensus calling and minimizes the impact of random error or rare contaminant reads. Adapter trimming and strict mapping quality thresholds were enforced during bioinformatic processing. Damage profiling using MapDamage2 confirmed the expected C to T substitutions at the ends of DNA fragments and fragment length distributions centered around 45 base pairs, matching ancient DNA benchmarks. To assess purity, the team applied multiple independent contamination checks. Nuclear contamination was estimated using X chromosome heterozygosity in this male sample, as well as the ANGSD in Schmutzi pipelines. Mitochondrial contamination was measured with the ContamMix tool. All estimates converged on less than 0.3% contamination, a level considered negligible for ancient genomics. The combination of clean room extraction, UDG half treatment, deep sequencing, and multi-layered authentication established this dataset as a new gold standard for ancient human genomes. The 2023 genome eliminates doubts about modern DNA interference and provides a secure foundation for downstream analyses of ancestry, phenotype, and lineage. Population genetic analysis of the 2023 high-coverage genome replaced earlier assumptions about Otzi's origins with precise figures. Using QPADM modeling, researchers calculated that 89% of Otzi's ancestry derives from Anatolian Neolithic farmers, with the remaining 11% linked to Western hunter-gatherers. No steppe ancestry was detected in any statistical model, contradicting the earlier contamination-affected genome. This ancestry profile situates Otzi before the major population movements from the Eurasian steppe that reshaped Central Europe during the Bronze Age. The Anatolian farmer signal aligns with archaeological evidence for Neolithic migration into the Alps, while the low hunter-gatherer proportion reflects limited local admixture. The QPADM results were cross-validated with principal component analysis and admixture clustering, both placing Otzi among Copper Age individuals with strong Mediterranean farming ancestry. Radiocarbon dating of the remains to around 5,300 years before present confirms that Otzi lived centuries before the arrival of steppe-derived groups in the region. The absence of steppe markers in his genome provides a clear genetic boundary between pre- and post-Bronze Age European populations. This corrected ancestry profile not only resolves the artifacts introduced by modern contamination in the 2012 draft, but also clarifies why certain genetic lineages, such as his paternal GL91 haplogroup, could persist in isolated alpine valleys. The 2023 data establish Otzi as a representative of the last wave of Neolithic farmers in the Alps, preserving a genetic legacy that was later diluted by incoming populations. These findings anchor Otzi's genome as a reference point for understanding population structure in prehistoric Europe, setting the stage for tracing his paternal and maternal lineages into the present. The 2023 high-coverage genome provides a detailed genetic portrait of Otzi's physical traits and health. Analysis of key SNPs linked to pigmentation shows that Otzi carried the ancestral alleles at SLC24A5, RS14266654A, and SLC45A2, RS16891982G, which are known to produce darker skin tones in modern populations. This pattern is consistent with the mummy's preserved tissue, and it contradicts earlier reconstructions that depicted lighter skin. For eye color, the HERC2 gene at RS12913-832 carries the ancestral G allele, a variant associated with brown eyes. No derived alleles for blue or green eyes were detected. Genetic markers for hair patterning reveal that Otzi possessed the C allele at RS1160312 in the androgen receptor gene, a variant associated with an increased risk of male pattern baldness. This matches the observed thinning of scalp hair on the mummy. The MC1R gene, which can indicate red hair, showed only the wild-type allele, excluding that phenotype. Lactase persistence testing focused on the LCT gene at RS4988235. Otzi's genotype was CC, the non-persistent variant, confirming lactose intolerance. This finding aligns with the broader pattern in early European farmers, who generally lacked the ability to digest milk as adults. Cardiovascular risk was evaluated through the 9p21 locus, RS1075728, where Otzi carried the G risk allele, linked to a higher probability of atherosclerosis. This genetic predisposition is supported by CT scans of his arteries, which revealed calcified plaques. The APOE and PCSK9 loci did not show protective variants, leaving his baseline risks unmitigated by genetic factors. Metagenomic analysis identified fragments of Borrelia burgdorferi DNA, the bacterium responsible for Lyme disease, in his bone. This direct evidence confirms he was infected at the time of death. Together, these results construct a precise image. Otzi had dark skin, brown eyes, a strong genetic risk for baldness, was lactose intolerant, had elevated cardiovascular risk, and suffered from Lyme disease. Each trait is grounded in high-confidence genotype calls from the 2023 cell genomics study, DOI:10.1016/J.xGN.2023.100377. In 2013, forensic geneticist Walter Parson and colleagues conducted a large-scale Y chromosome screening across the Tyrolean Alps. Their study sampled approximately 3,700 men, using a panel of around 500 SNPS and 30 STRS to identify fine-scale paternal lineages. Among these, 19 individuals were found to carry the derived G-L, 91 marker, the same Y, chromosome haplogroup as Otzi. G-L. 91 is a rare branch of haplogroup G2A2B, with its highest modern frequencies concentrated in alpine valleys and Sardinia, and only sporadic appearances elsewhere in Europe. The team calculated the most recent common paternal ancestor between Otzi and these men at roughly 10,000 to 12,000 years ago, based on calibrated mutation rates. This places the shared ancestor in the early Neolithic, long before the Copper Age. Mutation analysis showed just 1 to 3 private SNPS and 2 to 5 STR steps separating the modern carriers from Otzi, supporting a scenario of deep but continuous lineage. Using a Bayesian kinship model that factored in observed STR variants, population size, and mutation rates, Parson estimated a probability of about 60% that at least one of the 19 men descends from Otzi through an unbroken male line. The remaining probability accounts for collateral relatives who share the same ancient ancestor, but not a direct father-to-son descent. This evidence demonstrates that Otzi's paternal line persists in the region, surviving major demographic shifts that followed the Neolithic. The GL91 continuity provides a partial answer to the question of living relatives, although it does not identify any single individual as a direct descendant. The maternal lineage, with its own distinct history, would present a different pattern of survival. Mitochondrial DNA analysis once suggested that Otzi's maternal lineage, K1F, had vanished from the modern gene pool. Early surveys of tens of thousands of mitochondrial genomes found no living carriers of this rare subclade, reinforcing the view that K1F went extinct after the Copper Age. However, in 2026, FamilyTreeDNA's MitoTree project updated its global haplotype database to include a new match for K1F. The match was identified in Hedi Abad, a man from the Shawiya Berber community in northeastern Algeria. His full sequence. Mitochondrial genome was found to be either identical or separated by a single private mutation from Otzi's K1F motif. Phylogenetic analysis placed Abad's haplotype directly on the K1F branch, with a most recent common maternal ancestor estimated at roughly 7,000 years ago. This discovery was confirmed through triangulation with other testers from the Maghreb and southern Europe, who fell on adjacent branches, indicating that K1F survived at extremely low frequency outside the Alps. The presence of K1F in both a 5,300-year-old alpine mummy and a living North African individual points to a trans-Mediterranean migration corridor during the Neolithic and Bronze Age. The 2026 MitoTree update not only overturned the long-held belief in the extinction of Otzi's maternal lineage, but also mapped a direct genetic link between ancient alpine populations and present-day Berber groups. This finding completes the picture of Otzi's legacy. His paternal GL91 lineage persists in alpine men, while his maternal K1F line, once thought lost, endures in North Africa through a rare living descendant. Anatolian Neolithic farmers reached Europe not by a single land route. They traveled along a maritime corridor that spanned the Mediterranean. Ancient DNA studies show these early agriculturalists moved by sea from Anatolia, settling first in the Aegean, then spreading west to southern Italy, Sardinia, Iberia, and North Africa. Genome-wide modeling, including qPADM analysis, confirms that Neolithic Maghreb populations receive more than 70% of their ancestry from these Anatolian farmer sources. The survival of Otzi's rare GL91Y chromosome in alpine valleys and his K1F mitochondrial lineage in a Shawiya Berber community fits this pattern of Neolithic dispersal. Both lineages are relics of the same maritime expansion, preserved in isolated pockets after later migrations reshaped Europe's gene pool. The genetic matches, identified in modern Tyrolean men and North African Berbers, do not indicate direct, unbroken descent from Otzi himself. Instead, they point to a common ancestry shared with many other descendants of Neolithic farmers who traveled the Mediterranean corridor. The evidence from high-coverage genomes, haplogroup distributions, and ancient DNA from the Maghreb all support a scenario where Otzi's paternal and maternal lines are collateral branches of a broader family tree rooted in the Neolithic, not a direct line of descent. This synthesis unites the genomic data with the archaeological record, providing a comprehensive explanation for the distribution of Otzi's lineages in the present day. Modern contamination once obscured Otzi's genome. High-coverage sequencing corrected this, revealing 90% Anatolian farmer ancestry and erasing false step signals. Genetic variants indicate he had dark skin and a risk of male pattern baldness. Today, 19 Tyrolean men share his GL91 Y chromosome, while maternal haplogroup K1F, long thought extinct, persists in Algerian lineages, verified by FamilyTreeDNA and published in the journal Cell Genomics, DOI 10.101/J.xgn.2023.100377. Ancient DNA now connects prehistory directly to living people. What will the next genome reveal?